I don't know if there's anyone who does not know Richard Silverman in this session and in this symposium. He is, in my opinion, one of the best medicinal chemists in the world. He actually published more than 370 publications. He has 100 patents. He's an inventor, and he's also the inventor of Lyrica. We are thrilled to have him as a faculty at Northwestern and also joining our Les Turner Center as a faculty. And he's my long-term collaborator. Dr. Silverman is a Patrick Ryan Professor in the Department of Chemistry and Department of Molecular Sciences, Biosciences, and Department of Pharmacology at Feinberg School of Medicine. His research is focused on drug design, synthesis, and evaluation of small molecules, compounds, and that turn into drugs that inhibit enzymes, cancer, and protein aggregation. So some of those target enzymes include GABA aminotransferase for the treatment of epilepsy addiction, and he has numerous other compounds that are in clinical trial right now. And his work, especially on protein aggregation inhibitors, has received a lot of recognition, and I'm very thrilled to introduce him to you today. And when I asked Dr. Silverman if he could share a photograph with me from his childhood and a story, he shared this photograph, and he told me that when he was 13 years old, his brother and Dr. Silverman carried out their first chemistry experiment, the invisible flame experiment. The purpose of the experiment was to show that the clean burning flame was invisible, actually blue. And unfortunately, they used their bedroom as their laboratory. And of course, indeed, the alcohol burner flame was invisible, and the curtains were on fire. Can you imagine? The mother came home, came to the room and pulled down the curtain, put out the fire, and took the chemistry set away. So he was not allowed to have it back until he was 13. But then for 5 years of thinking how cool chemistry was, he was hooked on becoming a chemist. And recently, actually, he received a Therahedron Prize for his creativity. And I'm thrilled to introduce Dr. Silverman to you. And we are happy to be one of his colleagues here at Northwestern. Thank you so much. And Dr. Silverman, please take it away.
Thanks very much for the introduction. I learned a whole lot from that first experiment where I became a chemist by not allowing to have a chemistry set. And I've taken that on to my children, of course. I give you advice here for future parents. When you want your children to do something, tell them they can't do it. And then, of course, they do. And that seems to work most of the time. So today, what I want to tell you about, I do have a cold that I'm trying to fight over, but I want to tell you about this molecule NU-9 that we designed for inhibition of protein aggregation and potential treatment for upper motor neuron diseases, including ALS. I think I'll start by telling you the approach that we took to identify NU-9, the pharmacological and toxicological properties of the molecule, and then we'll get into the in vivo studies with Hande's lab and its potential in future treatment. So protein aggregation is really the hallmark for all neurodegenerative diseases, including ALS. The upper motor neuron diseases also have this protein aggregation: ALS, primary lateral sclerosis, hereditary spastic paraplegia, and as we heard today, spinal cord injuries. So very much connected to this. So ALS, as you all know, is premature degeneration and death of the motor neurons. Appears 10 to 15% is hereditary, and a quarter of those hereditary patients have a mutation in an enzyme called peroxide dismutase 1, or SOD1. And there's the animal model for looking at molecules to treat ALS is a model that has the mutation in this SOD1 gene. Mutation in the SOD1 gene causes the aggregates, and the aggregates then lead to the neurodegeneration. There are only 2 FDA-approved drugs for ALS right now, riluzole and darabont. So the way we went about it was by setting up 2 high-throughput screens, one to look for aggregation caused by PC12 cells expressing mutant SOD1, and then the toxicity resulting from this aggregation. Then we screened 50,000 or so compounds to look for those that might have an effect on inhibiting that protein aggregation. And as a result of that study, we found 17 different hit series that were important. We then filtered these series based on a bunch of computational methods and honed it down to 3 lead series. And these lead series were active in both of those assays. Today, I want to focus just on one of those 3 series, the one that led to NU-9, and we'll see how that happened. So for the chemists in the audience, that series called cyclohexane diones. That's a cyclohexane dione. It really is in equilibrium with something similar to it. But these molecules all had the second ring and something attached to it. And we had about 50 of these different molecules, 10 of which were active. And so we thought that this would be a good direction to go. We just needed to figure out what kinds of changes we should make. And of those 50 compounds, the best of them was this one with just a simple fluorine attached here. It had all the properties you want, except it really didn't have any good potency, which of course you don't want. But its solubility, its stability, metabolic stability was great. It crossed membranes very well. But we found that was a common feature to the series, is they had these pharmacokinetics, adenine properties, but poor potency. And so as a medicinal chemist, then we can go in, modify the structure, and then see if we can enhance the potency. And we did typical medicinal chemistry approaches, looking at electronic effects and steric effects, positional effects. And we tried to see if multiple halogens were better. And it seemed that if you put 2 of them on at these 2 positions, it was always better than other arrangements. And same thing here is with chlorine. That was better than the others. So that position seemed to be important. We found that you also, as you increase the size of this thing, it also seemed to be better. And so there must be something that this thing bound to that was larger. But as you put these larger groups on here and then attach a second one, the molecule becomes very insoluble and you really can't use it. So we had to then hone back on these structures. And what we found is when you put a trifluoromethyl group at each position in here, that then broke the micromolar, this is now sub-micromolar potency, which at least was our best compound that we wanted to proceed with. And it retained all the other good properties of the molecule, in addition to being much better in terms of its potency. So we carried out some off-target effects, things that are important to drug development. You don't want your molecule to interfere with this hERG channel, which is a potassium channel in the heart that regulates the rhythm of the heart. And so this compound does not interfere with that. And then we had it tested at this company for 68 different important pharmacological targets to see if we hit any of those, and it didn't bother any of those. So this seemed to be having some selectivity for whatever it is this thing is interacting with. And I should point out, we're looking for protein aggregation, which is a phenotypic screen. We don't know what the target is. We just know that whatever it is, it's inhibiting protein aggregation. So some animal pharmacology, we found that the maximum tolerated dose, this is how much compound you can give before the animal dies. What we have is 75 mg per kg. At low dose, it gets into the brain very well. But we were surprised then when we ran this ALS mouse model, the one we said that has a mutation in the SOD1 gene, it showed no activity. And it was like, wow, this thing had the properties that we were looking for. Why didn't this work? And you might say then, well, this is no good. You should have just thrown away this whole project. But I said we had 3 lead series. The other 2 were active in this. We were just wondering why this series wasn't active. And so we compared this molecule to one of the others in the other series that was active. Everything seemed to be similar in terms of its stability, half-life stability. Getting into the brain was about the same for the 2. But the big difference seemed to be this volume of distribution, which is, does the molecule distribute through the cells? And it seems that it's much lower with this molecule than one that was active. And so to us, that indicated maybe it's getting into the brain, it's just not getting to the neuron or getting to the right part of the brain that would be important for this. And so we looked at other cell types to see, does it get taken up into those? And we found, compared to this other one that was active and does get into all of these cell types, this molecule does not. It doesn't get into any of the others except for PC12 cells, which was the cell line we used in the high throughput screen. So it looks like it's not getting to the right parts of the brain. And more importantly, does it get into the neurons? And the answer again is no. Doesn't get into cortical neurons, whereas this molecule does. And so that seemed to be the problem. What could we do then to the structure here to help it not only get into the brain, but get into the right compartments in the brain? And so I had a very talented postdoc, Wei Zhang, who thought, well, maybe these 2 rings are too rigid here together, it's just held together, that we should put some flexibility in. And she made a variety of analogs, and having this kind of structure and this one did in fact allow them to get into cortical neurons. We then honed in on this molecule. Again, the one that was best had the 2 trifluoromethyl groups, but this flexible linker. And this one had good pharmacokinetics. For the chemist, this is a stereogenic center, so you have 2 isomers of that same molecule, and it's this isomer that has the greater potency than that one. But both of them get into cortical neurons, and so we then thought that this is the one we should go further with, and that turns out to be NU-9. So let me tell you about the properties of NU-9. It's quite stable in plasma, stable to microsomes, so that's metabolic decomposition. It's fairly stable. Gets into brain quite well, and it's orally active. So you can take this as a pill. 94% orally active means it's really getting absorbed quite well. It does cross membranes. Again, this is an in vitro study to show that it crosses membranes, but it doesn't then get effluxed out, which is important for a drug that you want to get into the brain and stay into the brain until it actually functions. It's not metabolized hardly at all. This was the only metabolite, and you can barely see it. So it's not getting degraded to a bunch of things that might cause some off-target effects. Other off-target issues, you can have drug-drug interactions when you have your drug inhibits or activates these cytochrome P450 enzymes, which are important to metabolism. So if you inhibit one of these isoforms of cytochrome P450, and that's responsible for the metabolism of another drug you're taking, so you can't metabolize it and it accumulates in your body, it can cause side effects. And so you don't want to interfere with these metabolizing enzymes. And we found that this NU-9 does not interfere with them. And again, it doesn't interfere with the hERG channel. We then did some preliminary toxicology in mice, and you give it's orally oral gavage. You're actually putting it into the throat of the mouse, and we kept increasing the dose because we were looking for some kind of adverse effect. And we couldn't see it at that 75 mg per kg dose, or at 90 or 300, 600, not even at 1200 mg per kg could we see any adverse effects, either externally or internally. And the animals seemed to be doing well, eating and body weight gain as usual. So it seemed like it wasn't having much of an effect at all. Then we did what's called a 7-day repeat, where you administer it orally each day for 7 days, and then follow and see what happens. And nothing happened for the first couple of days, but day 3, we started to see some adverse effects. And so we cut back on the dose from 1200 to 600, and then 300. When we got to 300, there were hardly no abnormal effects. There was a couple minor things that really aren't that important in humans. But then when you got to 100, there were nothing you could see different from the control wild type animals. So it's got a fairly high no observed adverse effect level. And it does, in fact, extend the life of the ALS mouse. Okay, so now we want to get more into how does NU-9 work in its effects. And I told you about this ALS mouse model, where what you're looking for is extension of the lifespan of this mouse by treatment. And unfortunately, there's no correlation between what you find in this mouse model and what happens when the molecule then goes to clinical trials. But this is the mouse model for ALS, and this is what everybody uses, but it seems to really not be related to anything that you could use in therapy. And maybe the reason is the extension of lifespan of the mouse has nothing to do with human ALS. It's some other cause. So I teamed up with Hande Oostendler, who had been for quite a number of years studying upper motor neurons. And she and others have found that the upper motor neurons, these are the corticospinal motor neurons, start to degenerate very early in this disease, actually before symptoms occur, both in animal and in human cells. And so since the upper motor neurons in mice and in humans are really functionally and structurally quite similar, it seems that if you could have a positive effect on upper motor neurons in mice, that should translate to humans. And so this could be a new paradigm then for looking for treatments in upper motor neuron disease and in ALS. The problem, though, is you want to look at what's happening in the upper motor neuron. And that's tough because there are a couple billion neurons and only a couple 100,000 upper motor neurons. So how are you going to even see an upper motor neuron in the presence of all these other neurons? Well, Hande figured that one out. She found that the upper motor neurons express in large amounts an enzyme that other neurons do not. And so she was able then to express this fluorescent, green fluorescent protein with this enzyme. And so now she had a fluorescently labeled upper motor neuron. And the fluorescently labeled ones, the one that has this green fluorescent protein attached, doesn't seem to have an effect on the upper motor neuron itself. And so then she took the mice that had these fluorescently labeled upper motor neurons and crossed them with ALS mouse models. The mutant SOD1 model, she's now done it with TDP43. She's done it with profilin, alsin. She has quite a number of these, but you cross these and then you end up with a mouse model that has fluorescently labeled upper motor neurons that we can then study with our compound. And we said these upper motor neurons are pretty much the same in mice and in humans. So here's what a healthy and an unhealthy upper motor neuron looks like. When they're healthy, they've got these long axons and stable cell bodies. But the unhealthy ones have got truncated axons. They're starting to degenerate and the cell bodies are degenerating. So these are very poor health. But if you treat these neurons that are taken out of the mice that have mutant SOD1 and then treat these in culture with our molecule, you see that the axons remain long, the cell bodies are stable. And so if you, the upper motor neurons that weren't treated with this degenerated in a rapid period of time. And in fact, the axon lengths were increased relative to the animals that had a mutant gene. So here are the wild type animals, and that's the axon length. These are the mutant SOD1 animals. You can see their axon lengths are much shorter. Treated with our compound, the axon lengths are at least as long as wild type. But if you treat them with those two FDA-approved drugs, riluzole or edaravone, again, there's not an increase back to wild type. But what we did find, or I should say what Hande and her group found, is that if you use these in combination, then you get an enhanced effect. And so here's that same study, but she also included a drug that's in clinical trials, AMX0035. And again, that one axon length doesn't increase as much. But if you now combine NU-9 with riluzole, see the axon length shoots way up, with edaravone shoots up. But with this experimental drug, it really doesn't change when you give NU-9. And so maybe these are multiple mechanisms going on, and we're enhancing these by having two mechanisms acting together. So now let's take a look at the in vivo data that Hande got with this molecule. And so here is the general health of upper motor neurons, and we're looking at the number of healthy upper motor neurons in the wild type animal right here. This is the animal that has the mutant SOD1. You see it's very unhealthy, but we showed in a dose-dependent fashion, 20 mg per kg and 100 mg per kg of our compound brings it back almost to that of, well, it overlaps, so it's pretty much the same as the wild type. Apical dendrite degeneration is common also in ALS, and you can start seeing that they're degrading vacuoles are formed. And so you can measure the number of vacuoles. And here in the wild type, you have a relatively low number of vacuoles. The animals that have the mutant SOD1 have a large number of vacuoles, but here's 20 mg per kg and 100 mg per kg of our compound, again, bringing it in the range of the wild type. Protein aggregation, we said, was what we're trying to prevent And here's wild type, very little protein aggregation, but the mutant SOD1 animals, there's a lot of aggregation, 20 mg per kg of our compound less, 100 mg per kg, even less protein aggregation. And now we heard a lot today about mitochondria, endoplasmic reticulum, and how important that is to ALS. Here is the wild type animals, the cytoplasm, the mitochondria, looking at the inner mitochondrial membrane and the ER. And then this is the mutant animal, and you can see cytoplasm is falling apart and the mitochondria are falling apart, as well as the endoplasmic reticulum. But treatment with 100 mg per kg of our compound brings it pretty much back to wild type, which you can see here. This is the percent of healthy mitochondria in the wild type. This is the healthy mitochondria in the mutant SOD1 animal. And now this is after treatment with NU-9. The mitochondria are quite healthy. We also heard today how important TDP-43 is in this disease. And so we took a look at these same experiments in the mutant TDP-43 animal and got the same results. And so this is really exciting, particularly hearing how important TDP-43 is to the progression of this disease and to the frontotemporal dementia. Again, here is healthy mitochondria, wild type TDP, mutant TDP-43, and treatment with NU-9, and same with the ER. Treatment of NU-9 maintains the health. Same with the apical dendrites in the TDP-43 and upper motor neuron. There's wild type. There's the vacuoles in the mutant TDP-43, and here's with 100 mg per kg of NU-9. The upper motor neurons, this is the healthy upper motor neurons and wild type in the mutant, and then this is with 100 mg per kg. NU-9 brings it back to that wild type level. And well, this is an in vitro study, but again, it's same with the axon length, that it increases the axon length of the mutant SOD1 TDP-43 animal, even larger than the length of the wild type. And while all this was going on, there's no change or no decrease in the body weight, which is usually an indication that the animal is feeling okay and is relatively healthy. So these are the non-mutated animals, and the mutated animals, doesn't matter, the wild type and the treated are the same here. And then finally, a behavioral study was done where you attach the mouse grips onto this mesh, and then you invert it. So the mouse has to hold on. And this is a measure of grip strength, which is an upper motor neuron issue. And what you find is the animals, as they proceed in the disease after P95, they start losing the grip, and they can hold on less and less as time goes on. Whereas the animals that were given NU-9, both the SOD1 and the TDP-43 retain their grip strength. So in conclusion, we started with a phenotypic screen to identify inhibitors of protein aggregation that arose from mutant SOD1. So we were hoping to get a molecule that then would be important to inhibition of protein aggregation in general, not just from that caused by mutant SOD1. And sure enough, we've been able to show that it works with also mutant TDP-43 And Dr. Osdiller has a couple other models we can work on. I have some preliminary data supporting its effect in Alzheimer's disease as well. Again, another protein aggregation. So we're hoping this could be a paradigm shift in how you look for molecules to treat upper motor neuron disease resulting from protein aggregation. And last year I started a company called GABA Therapeutics, and we're now in IND enabling studies for NU-9. For those that are following NU-9, it's been renamed as AKV-9 for this company. So you may start seeing something with that title. Of course, I need to thank the people that did this. I just sit at home blowing my nose and feeling awful while they do all the work. And quite a number of students here that have worked in my lab, the students in Hyundai's lab. Rick Morimoto was the person who developed actually the screen for this protein aggregation. This is the startup company, Cambria, that did the high throughput screen. And then Bob Ferranti did some of the earlier toxicology. And of course, we have to thank the organizations, the ALS Association, Northwestern, and NIH for supporting this research. And we thank the organizers for inviting me to present this, and thank you very much for your attention.
Thank you so much, Dr. Thank you so much, Dr. Silverman.
Stop sharing. Go ahead.
Thank you so much for your presentation. It's unfortunate that, we're not in the room. We can't all applaud, and, so you don't know how much your presentation is appreciated, but I can tell you from the questions, and I can tell you from the participation that people are really interested in NU-9, and they would like to learn more. Especially they would like to know how it is going and how clinical trials, if there is any work towards clinical trials, they're interested to see if they would be included in those clinical trials. And I'm sure most of those questions will also be answered in the clinical panel as well. And, what makes NU-9 different from other compounds in terms of ALS? But maybe I should read questions one by one, so it will be easier for you to tackle them. It says, amazing work with NU-9, Sarah McDonald. And it says, this gives me reason for optimism. I know patients and families will be all anxious to start clinical trials. Do you envision when this could be?
Yeah, well, as I say, we have to go through a lot of IND enabling studies, including safety studies, a lot of toxicology. We're synthesizing a large batch of the compound through a CRO to do these IND enabling toxicology studies. And then, writing the IND and submitting it. This is going to be at least a year, could be a year and a half before it's really ready for clinical trials. And I think we're probably going to have to do a few months of healthy volunteers to begin with. And so the patients then won't get this closer to a year and a half, I would think.
Yes, the first phase 1 would be on non-patient population. So even the phase 1 would start without the patients, but the patients would have phase 2. Mm-hmm. So Christopher Southen asks, how does the structure activity relationship, the SAR of NU-9, look like in animal studies?
Well, we didn't do a structure activity relationship in animals. We only did it in the cells. And, that's how we honed in. It was about 4 years of chemical modification to get to NU-9 based from that first molecule. I made it sound that we did it overnight, but based on that first molecule that had the two rings attached to get to that flexible one with the right groups and all. I said it was about 4 years. And throughout that whole process, we were able to improve the activity. But you know, in a academic lab, we can't afford to do animal studies with every molecule. And in industry, they start doing animal studies with a lot of different molecules.
Yes, but you have done it in the in vitro models, right?
Yeah.
Okay. So it says, what is the molecular mechanism of NU-9? Do they target or interact with distinct set of proteins, or do they target a wide array of proteins or molecules?
Yeah. Well, a few years ago, we started to do those studies, but I said we had 3 different series. And I guess, unfortunately, now we picked a different series to do those studies. And we now know the target for a different series that we made. And it is, in fact, a single target. We are now starting, actually in conjunction with NIH, studies to do the same with NU-9. And so I'm hoping in the next year or so, we'll have an answer as to what's the specific target other than the effects of whatever it's interacting with.
Yeah. And because it improves the mitochondrial inner membrane, it may also be not one protein, but cardiolipins or proteoglycans or other molecules as well, correct?
It could be. Okay.
All right, so now Christine Moretti says, so grateful for the work and for your presentation. As a PLS patient, I'm truly excited for NU-9. Thank you to everyone for your commitment and dedication, she says. And Peter Werner asks, in your opinion, why wasn't an additive effect of NU-9 with Emilix 0035?
Oh, yeah, I don't know. Because I mean, I don't know what the mechanism of that. It's a drug combination. I don't know what that does, but apparently it's not additive or synergistic with NU-9. You know?
Yeah, so if I may add to that, so the Emilix compound is actually a combination of 2 different compounds which have been previously FDA approved, and those are important for mitochondrial stability and ER stress. So they created a cocktail compound. So the Emilix is a cocktail for 2 compounds. So it looks like the NU-9 improves the health of mitochondria. It also improves ER integrity and protein aggregation. It seems they may be working on a similar pathway. So if that's the case, we may not see an additive effect, but the riluzole, as you know, reduces astrogliosis, and other one is free radical scavenger. So there are 2 different paths, and this may be the reason why we see additive effect. But of course, I think more needs to be done, right? So Dr. Piora asks, does NU-9 have neuroprotective effect on lower motor neurons, if less than its effect on upper motor neurons?
Doesn't seem to. I think they did look at that, and it does not. But our hope is that if you protect the upper motor neurons, that you won't lose the connectivity to the lower motor neurons, and it could also then have an effect on maintaining the health of the lower motor neurons, and particularly in spinal cord injury, where if you can prevent that disconnect from occurring, then you wouldn't have the degeneration of the lower motor neurons in spinal cord injury.
That's right. And then Dr. Piora asks, how did you decide to test NU-9 on upper motor neurons, since the screening process of the compound was actually based on a non-neuronal cell?
Thanks to Dr. Oostendler. She had these studies that seemed to indicate their importance in ALS, and we wanted to see if this is the direction that this molecule was taking.
Yes. So yes, I should take some blame for that one, because when the NU-9 was identified as a compound that reduces protein aggregation and improved neuron health or cellular health, we knew that this problem occurs also in the upper motor neurons, and we had a reporter line. So we wanted to see if that also works in diseased upper motor neurons, which degenerate due to similar underlying causes. All right, so wish that given absence of effective therapies for this disease, that promising drugs could be given to willing patients' consent with less rigorous testing and safety studies. I'm not sure if that's possible, and I'm sure we're going to discuss this more in the clinical session, but I don't think it is possible to give drugs to patients without we complete the clinical trials. What do you say, Dr. Silverman?
Yeah, I really don't want to go to prison before we see if this works. And so,
yes.
And also you don't want the study not to be done properly, and it's not working. And so we want to do it the right way and have it done through the right path.
Yes.
But I've had a number of patients write to me and say, can you just send me some of this and I'll take it? And it's like, I really would like to, but I can't do that.
All right, so I think that includes all questions and answers. If there's any other question that I didn't see. All right, so does NU-9 reduce SOD1 or TDP protein aggregation? I think you answered that already. Yes, there is. Yes. Okay, so I think this will then end the science session with Dr. Silverman's excellent presentation. So I would like to thank all presenters for the morning today. And as you have seen, we have a wide range of speakers and topics. And because ALS is also very complex, we need to approach ALS from many different angles. So iPSC cells are very important for understanding the cellular mechanism. And we hear from Dr. Ward, they're also important for the identification of biomarkers, especially within the context of TDP pathology. And Dr. Jane Wu has told us, again, within the same context of TDP pathology, the importance of mitochondria, mitochondrial defects, and how do they contribute to motor neuron degeneration, both in different model systems as well as in human motor neurons. And Dr. Franz has showed us the imaging technologies and the novel innovative approaches that he has developed in his lab for developing outcome measures for future clinical trials, and how the corticospinal tract degeneration and injury also contributes to ALS. And with Dr. Silverman's talk, we discussed the identification of NU-9, a novel compound that improves the health of diseased upper motor neurons. And even though ALS is characterized by degeneration of both upper and lower motor neurons, the cortical component has been ignored for many years. And we hope to bring everything together and develop effective and long-term treatment strategies where we actually find therapies both for the cortex and the spinal cord. And I would like to thank everyone for their excellent participation, the questions, answers, and for all your attention. I am very thankful to you.